Hey everybody, so I'm introducing Raymond Walters. Yeah.
I'll let you be further away.
Okay. So I worked with Raymond at the Broad Institute when I was a postdoc. Raymond's currently a researcher at Mass General Hospital and a group leader at the Broad Institute. He got his PhD. Ooh, yeah, I know this. Duke. No, not Duke.
Nope.
Catholic Notre Dame. He got his PhD from Notre Dame in psychology, right?
Mm-hmm.
So he's brilliant and nice and knows all about open score regression. And so, there you go.
Thank you, Patrick. So yeah, gonna spend some time today and continuing into tomorrow talking about this lovely method called LD square regression. That's been hyped at least a little bit in the talks you've heard already. And so we've
Raymond, sorry, no sound. Can you unmute yourself?
Just up at the top. Oh,
Yeah, it doesn't show in the presenter view. Now we good?
But now we see presenter view.
Oh,
here.
now we see. Now we
Does it
good.
look right
Are we
for
going on
people
audio?
Zoom? Yes.
It looks right, yeah.
Great.
Cool. So yeah, going to spend some time talking about this method called LD square regression, which does a few different things. And so the piece we're talking about today is around heritability and partitioning heritability, and also kind of the estimation of stratification that comes with that. And so just to there we go. So just to fully motivate this, kind of a little bit beyond what's already been forecasted by the previous talks, why are we spending this time talking about LD square regression? Well, there's both the kind of the direct utility as far as why it's a useful method, and also just its amount of use in the field. So as a method, it's a computationally convenient way of getting at questions about heritability, stratification, and genetic correlation or covariance from GWAS data, which are potentially parameters of interest in their own right. So we've talked plenty already about kind of this idea of heritability, and this idea of overlap between different traits in genetic spaces come up, and certainly concerns about wanting to be aware of stratification in our results. So being able to estimate those quantities is certainly of use. And in particular, having computationally convenient ways to do that. So there's a range of estimation methods, but LD score perhaps has this, by way of some shortcuts on the assumptions you make or some losses of statistical efficiency, is much less computationally demanding than some of the alternatives that would use individual level genetic data as opposed to LD square regression being able to work just from GWAS summary statistics. And the other kind of utility of talking about it, even if you walk away after hearing all the caveats that I've had to throw in about the interpretation of LD square regression results themselves, is that kind of despite that it's widely used and increasingly is kind of a foundation upon which other statistical methods in the GWAS space have been built. So some of which you'll be hearing more about next week in this course. And so knowing what kind of foundation those other methods are building upon is potentially of utility, even if the takeaway from this turns into that as a SNP heritability estimation method, there are much better alternatives than just plain LD square regression. And so to avoid backloading those limitations too much, the things we're going to flag in terms of those limitations is that there are indeed other more efficient estimators for at least most of the quantities that LD square provides, in particular for SNP heritability, where the kind of the trade-off for being such a computationally convenient method and being able to work with summary statistics is that you do pay a cost there for not taking advantage of the better information available if you had the individual level genetic data. The other thing is a lot of that simplicity of calculation is built off of making simplifying assumptions in the modeling. And so in varying cases, those assumptions may just not be viable. And kind of the last thing I'll flag here is that, you know, I'm going to spend a lot of time on technical detail of how we get to an estimate of SNP heritability or to an estimate of genetic correlation. But there are many scientific questions for which that's a stepping stone or just not terribly informative to our actual questions of interest. And so despite the fact that depending on what part of the GWAS literature you're looking at and what exact slices of years, it seems to have become a de facto norm that at least most large GWASs will include somewhere amongst their results a description of LD score regression results. To what extent that's actually informative beyond some kind of pro forma checks, and how much we may or may not care about, say, a SNP heritability result, is kind of an open question.
Yeah. In the first bullet, by efficient, do you mean in the statistical sense or in the computational sense?
In the statistical sense, so in terms of width of the standard errors on our estimates. Depending on what set of things you want to consider competitors, you could also say in the computational sense. So it is faster to compute a twin heritability off of just those correlations than it is to crank through the necessary set of things for the GWAS-based estimate from LD squared regression. But in any case. So pretty much everything we'll talk about in terms of technical detail of kind of the original core of LD squared regression goes back to these series of 3 papers co-led by Brendan Sullivan and Hilary Finucane, who were both graduate students at the time. There's been a number of extensions building on top of this, but the kind of core methods exist in these 3 papers, and we'll be walking through roughly in this order the kind of initial description of LD squared regression, then this partitioning idea, and then the genetic correlation idea. So let's start with the intuition out of that very first LD squared regression paper, which was, as has already been discussed, when we look at the results from a GWAS and we see our QQ plot has this inflation, there's room for concern, especially depending on how far down towards the bottom of the plot there you see this deviation from expectation, that we might have stratification present in our GWAS. But the alternative that could be claimed is that there's simply a high degree of polygenicity for our trait of interest. So maybe it's like height, and majority of things seem to have at least some degree of association with the trait, even if you've fully controlled for PCs and done your full faith best to correct the stratification the best you can. And so it'd be nice to be able to identify whether or not we're dealing with stratification. So the toy example here is actually that these are from simulation. One of these plots is simulated under there being a truth of just polygenicity, and the other is simulated under the truth of there being just stratification. And I'd be hard pressed to tell you just by eyeballing a QQ plot like this to tell you which is which. And so can we get to some statistical statement that will help us differentiate between these 2 scenarios? And the kind of motivating application when they wrote the original LD score paper was the schizophrenia result. I think you've already seen this Manhattan plot before, but again, it was kind of real result, fairly early deviation from the diagonal part of the QQ plot. And so was there some or all of this that was due to stratification rather than polygenicity? And so the intuition that went into LD score went something like this, which is that let's imagine our toy section of the genome here, where each of these tick marks is a different SNP that we have measured. As we've learned from the previous talks about the structure of the genome, there are blocks of LD here. So as a simplifying toy example, we'll say that within those blue boxes there, those sets of SNPs are all perfectly correlated with one another. So you've got the initial group, all those SNPs are perfectly correlated. You've got a couple of SNPs here that are between those blocks that aren't correlated with any of the other ones around them. And so this is our genetic structure. And then when it comes to thinking about how our toy genome here is affecting a trait, we can imagine that in some pseudo-random process of where polygenic effects arise in the genome, that a particular couple of SNPs here are causal for our polygenic trait. And so they land at these 2 particular SNPs in the genome. And so if that was the case, then what we'd expect to see, once we did a GWAS of this data, is that all of the SNPs in those corresponding LD blocks would be associated with the trait because they're in LD with the one true causal SNP that is within that block. And the SNPs that aren't part of those blocks would show no association. And we can imagine that if we were, simulating random traits or thinking about traits having arisen kind of at random, we could repeat this process again, throw down another couple causal SNPs for this new trait. Similarly, we'd see association of those the SNPs in their corresponding LD blocks to the trait, and so forth. And if we were to repeat this process over and over again, what we would expect to see on average is that your likelihood of showing association in a GWAS sense as a SNP in this toy genome would be proportional to the size of the LD block that you're in. So if you are a SNP that is in a large LD block and tags a lot of other SNPs, then under this simplifying version of having distributed the true causal SNPs kind of randomly across the genome, then your likelihood of being associated into GWAS from these kind of randomly distributed polygenic effects would track with how large an LD block you're in. And so this gives us, under what were a very simplified toy model version of what distribution of causality would look like, but at least an intuition for some relationship between LD and the degree of association seen in a GWAS under this kind of polygenic model. And so the counterfactual that we might consider is, all right, that was all with their true causal effects. But what if all we're seeing in a GWAS is the result of some random population drift that has confounded our GWAS by way of population stratification? And so this is a little harder to diagram the intuition, but I'll do my best to describe why these numbers are what they are here. But the idea is that to the extent that we observe differences between parts of a population or different populations, in terms of population structure, so like before we consider a phenotype and we just have like the PCA type observation, that kind of the unit of differentiation between those populations is these LD blocks or the haplotypes rather than the individual SNPs. So to the extent that these blocks have continued to travel together and are still showing LD across the population that we've observed, whether or not that particular block has diverged in frequency between a pair of populations or whatever dimensional space is populations. Those SNPs hung together, and so their likelihood of drifting is as a group rather than on an individual SNP basis. So if, in our toy example, there had, via the random walk of drift between populations, we had we especially expect uniformity of that probability of drift that's not related to the size of the LD block. Again, simplifying assumptions. In particular, we're kind of ignoring any possibility that there might be a relationship between LD and selective pressures that would then be reflected in drift. But under this kind of simplified model, we'd expect that under pure drift, we don't expect a relationship between LD and the degree of signal in the GWAS. And now that means that we've now said that if we have polygenicity, then there should be a relationship between the GWAS signal and LD. And if we have stratification, then we shouldn't see a relationship between GWAS signal and LD. And now, can we turn that into an actual quantitative statement? And so, but we're going to take some time to work through how we get to this equation. But where we're going is it turns out that under the simplifying set of model assumptions we're going to make, we can say that the expected chi-square statistic from our GWAS, right here, so that squared beta over standard error, is an expectation equal to a constant term that consists of a literally one and then a piece that scales with sample size that's related to stratification. We'll talk more in detail about what that is. And then a term here that is proportional to SNP heritability, scaled related to sample size and number of SNPs. And this term that we're going to define as our LD score, which is a sum of the amount of LD for a given SNP to essentially all of its neighboring SNPs. So this is where we're going, and I'll show you how we get there. The starting point is to go back to the same kind of additive genetic model that we've talked about a few different times. So we've got our phenotype that is related to some additive genetic effects of our genotypes, some confounding covariates, and residual here. And just for the version notation I have on the slides, we can do this in summation notation, where we're going to focus especially on these beta stars here, where we're going to use throughout here the notation of that star to try and distinguish between this beta is in this joint model. I'm not going to claim that these are the causal betas due to tagging of LD, or, of say rare variants that are outside a SNP set, or other structural variants if we're talking only about H square SNP and not kind of broader all of heritability. So just our kind of best linear predictor version for some universal SNPs. But in a joint model rather than our marginal GWAS betas where we've tested the SNPs individually. And for convenience, we've got this standardized, so this is the standardized version of the phenotype, and we standardize all the genotypes as well to unit variants. Mm-hmm. Where are we? That's just for to distinguish. So this is a sum over SNPs, and this is a sum over however many covariates you're dealing with. I think it's L here just because we're going to be talking about multiple sets of SNPs, so we're keeping K handy for talking about other sets of SNPs. Yeah, it's not. Right. Yeah, so this is not that LD score. I agree that that's confusing. I'm sorry for that. Yeah, no, this is purely for indexing L and is not at all related to this L. Cool. And so from our simple additive model, we recall how we have our definition of what our heritability is. And so with the normalization and pulling down the notation, we can say that the variance of the sum of the genetic effects is now equivalent to heritability, at least in the sense of a SNP heritability for the set of x's that we've put into this sum. Cool. So if that's our model, then it's a nice model, but what we've observed, at least for LD score purposes, is the set of betas out of a GWAS. And these are not those beta stars anymore. These are now simply for a given SNP, its covariance with the phenotype, which means that if we walk through from how we've already set our model for the phenotype and kind of divide up the sum there, that we have a sum of, for our given SNP, a sum of its correlation with other SNPs. So this is this R. So this is a correlation between our target SNP, SNP-J, and other SNPs around it, SNP-K, and the effect in the joint model of those other SNPs. So essentially, this is the extent to which SNP-J tags associations that are happening and the kind of joint effect size value of those effects that are being tagged. So this we might think about as our quote-unquote real GWAS beta, or at least the part that we're not worried about being confounded, as opposed to that we also have potential correlations between our SNP and other confounders. So for example, our ancestry piece would live here, and then we have residual error in our estimate of those GWAS betas. All right. So we've got that statement now about what our GWAS betas look like in relationship to these kind of underlying joint estimates of effect sizes with the consideration of stratification or other confounding that might be sneaking into those effect sizes. And so now is where we're going to make a bunch of assumptions that let us do the the nice things of LD score. So.
Sorry, I was a bit
Yep.
lost somewhere. What is subscript K?
Subscript K is is also for SNPs,
Oh.
and so our GWAS for SNP J relates to the relationship between SNP J and all other SNPs, SNP K, where functionally, assuming that LD remains local, is a set of SNPs that is local to that SNP, but in theory, model holds for genome-wide. I'll talk a little more about that windowing piece later. So, assumptions. Let's start by saying that those beta stars, those joint GWAS betas, are random. We're going to treat them as random effects, mean 0, with a variance that, for totally arbitrary reasons, we're going to say are equal to a SNP heritability over a number of SNPs. Just happens to work out. More importantly, we're going to say that not only are these random, but in expectation, there's no covariance between them for different SNPs. So if I could tell you that the kind of population true effect size for the SNP in the best linear predictor was a given value, it would tell you nothing about kind of neighboring or other locations in the genome, what those joint effects look like elsewhere. We're also going to say that there's no relationship between those effect sizes and the amount of LD. So this is what I was kind of getting at in that intuition. So at least in certain models of neutral drift, this is a fair assumption. But we're going to bake this in as an assumption that there's no link between the kind of size of your LD block and your causal or conditional joint effect size. So the only, well, it'll turn into that there's a relationship between that effect size and LD tagging in the marginal, but not in the conditional. And then similar random effects type assumptions on the stratification piece. Again, neutral with a variance parameter, and no correlation between either the stratification and the effect size or the stratification and our error. And so all those are assumptions, some more defensible than others, but they make our lives easier. And one other piece we're going to grab just from OLS is to say that the estimation error in our GWAS betas, so just this term, not including the stratification concerns, should have the standard behavior of OLS betas, where once we account for the fact that we're talking about having a normalized phenotype as our scale, should just be proportional to sample size. If we assume that we've normalized such that the effects of any individual SNP are small. So especially for highly polygenic things, we know the explanatory power of, say, any individual SNP for educational attainment is tiny. The difference that makes in the residual variance between normalizing the phenotype versus normalizing the individual is tiny enough that we can just get to only worrying about sample size when we look at the variance of that term. And so having made all those assumptions, if we go look at our chi-square statistics from our GWAS, we start to be able to move through to where I said we were going in terms of that expectation for the chi-square. And so by definition, our chi-square statistic in the GWAS for an individual SNP is that squared effect size over its estimated variance. And so we're just going to plug in what we've said is our expectation for that GWAS beta, and that variance has now disappeared via the 1/n term, is now living out here. Having made all those assumptions about orthogonality between the different pieces, this variance decomposes nicely into separate pieces. I reversed the order of the terms here, just for better alignment with how the LD score equation is normally represented as far as where we're going, but it's still the same terms. And we've just dropped the cross terms now that would otherwise appear out of expectation for that square. And so what we end up with is, so via our expectation for the variance of the error term, we get nicely out the 1 there from our OLS piece, via how we've chosen to define an expectation for the variance of the confounding piece. We now have that called this A here. And then for our summation over the tagged LD effects, because we've said that there's no relationship between the effect sizes and the degree of LD, we can extract this out. And we now just have expectation for this beta squared, which is our variance of that beta, and the inner term that is, again, that sum over LD to the set of SNPs that are SNP J is summing over LD to all other SNPs K around it. And so we're almost all the way home. So we had previously said that we're going to list the parameterization of this of the variance here in terms of this h squared over m. So here's where that is popped out. And we're just distributing terms through, and we're down to essentially where we're just going to plug in that by definition we are calling that sum of LD to other SNPs, the LD score of the SNP J there. And so plugging that in gets us to that formula that I said we were headed to, that we now have our expectation of our GOS chi-squares in terms of this variance component that lines up with SNP heritability with LD scores, and a constant term that is not varying with J there. That's just a flat A. Yeah?
What's M again?
So M is number of SNPs. So one way to think about kind of why we ended up with this labeling before, I'll go back to here where it's a little separate. Is that these are the conditional effect sizes, and so the variance of those conditional effect sizes, we're saying that's kind of existing on average in a random effect sense across the GWAS, is you have the kind of total amount of variance that's there divided by the number of SNPs that's been split up amongst randomly. And so this does work out that you get a total for the phenotype variance coming from the set of x's of this h square SNP. So it's divided amongst those m variants. Cool. We were here. Cool. So we made a lot of assumptions to get here, but we now have an equation. Does it work in our at least toy example here? So as I told you at the outset, these were simulated examples of one GWAS with nothing but stratification and one GWAS with nothing but polygenic effects. And so what we see is that for this top one, which is the stratification example, if we take all the SNPs that are here, bin them by their computed LD score, so that sum over the amount of LD to neighboring SNPs, and then look within each bin at the mean chi-square of that set of SNPs, that gets us each of these dots. So each of these dots is kind of collapsing a much larger cloud that exists vertically here. But that indeed, when we simulated with population stratification, then we see no relationship between those LD scores and our mean chi-square statistics out of this synthetic GWAS. Versus in this bottom scenario, where we did simulate with polygenic effects, we see this nice linear relationship between those kind of the mean chi-square in those bins of LD score. That lines up exactly with our expectation for what that slope should look like in the case of polygenicity. And gratifyingly, in that first set of applications of LD score regression, a number of GWASs were looked at to see how well they looked by this metric. And so this is what I think this was EA1 that this application was to at the time. Lined up nice and perfectly, nice linear relationship here. Things looking good for an explanation of polygenicity as opposed to concerns about stratification. And so that's a general statement about fit of the line, but what do we actually get in terms of estimating quantities of stratification and SNP heritability? Well, so what we've pieced together is that we have a thing we've observed here in our chi-square statistics, a thing we can at least compute here in terms of LD scores, so LD of the SNPs with their neighbors, and then a term that we say should be constant across SNPs, and a term that is essentially looking like a coefficient here. And so we've got all the material for a regression. So we can plug in our observed values and get an estimate of this intercept term here and of this coefficient on the LD scores that serves as a slope. And so both of these things are useful. The first is that for that intercept, how we arrived at the expectation for this constant is you've got a piece here that is kind of the irreducible expectation just from kind of the random variance. And if we think of the null distribution for a 1 degree of freedom chi-square statistic, that's 1. So kind of under the null, there is none of this other piece that scales with sample size. And then you just have that amount of error variation. But that A, we recall, came from the variance of that term that was our stratification bias confounding term, those S sub j's. And so this intercept gives us a way to look at deviation from an expectation that, regardless of whatever is being explained in terms of SNP heritability, is there another piece of stratification in that chi-square that is not explained by that relationship, and thus might suggest that there's some confounding existing in the GWAS. And then similarly, since we have this coefficient and we know sample size, and at least if we make some assumption about what M should be for our universe of SNPs that we're considering for our joint model, we can back out an estimate of the SNP heritability from that regression coefficient from having done this regression. The scaling for binary traits, we have to do an additional conversion because all this will exist on kind of observed scale of variation as opposed to thinking about liability scale effects that happen for loss of information from binarizing and from any ascertainment of, say, oversampling cases versus controls. But those are kind of solvable issues, and either way we can get to a SNP heritability estimate. So I just said that we have the things we need in terms of LD scores and chi-square statistics, so where are we actually going to get those LD scores? The ideal circumstances, in terms of what I've just walked you through for the derivation, is that we want in-sample LD for the set of individuals in the GWAS to avoid having to worry about how effect sizes behave on the sex chromosomes. We might focus on just autosomal LD and talk only about SNP heritability for autosomal SNPs. And because of the way we've treated covariates, presumably in the GWAS and in our modeling, what we really want is that LD residualized on those GWAS covariates. So in particular, controlling for PCs that reduce stratification that would otherwise show up as long-range LD between SNPs at a much longer distance, including between chromosomes across the genome. So in practice, if we've done that correctly, then we'd expect to be able to only worry about computing LD within those localized LD blocks. In practice, software generally looking at something like a 1 centimorgan region around the SNP in terms of where it's going to sum for that LD. And this idealized case does exist now a little bit in practice. It didn't last time I gave this talk. In a kind of extension called COVE LDSC, where that COVE is implying that use of the covariates. But I told you that part of the reason we like LD score is because we don't need that individual level data, and this sounds an awful lot like needing individual level data about our sample. And so more commonly, and the more frequent application here, is to instead use LD estimated from some population matched reference samples. So no, it says ease of access to the data. We expect it to be mostly right in terms of consistency of LD between some reference panel and some, well-matched, at least to the level of, say, continental ancestry. And empirically seems to behave quite well, but there is some slippage here in what we're saying about why this works. But in practice, for for a long time, it was just 1,000 genomes-based LD. There's been some movement in applications that we're seeing more use of LD from UK10K or HRC samples or other larger reference populations. But in any case, this gives us a way to make use of LD scores without access to the raw genetic data of our actual GWAS sample. And so the kind of the final point here is that if we had the raw data, we could do this, but also we could use some entirely different estimator for our SNP heritability that would have those efficiency gains from not relying just on GWAS summary statistics along with this, and would actually do modeling within the individuals themselves. So methods like GCTA or Gremel or Hazeman-Elston regression that would get us tighter standard errors and potentially more accurate estimation. Yeah.
Could you briefly explain the differences between Gremel and the LD score?
Ooh, to do that briefly. So the conceptually, yeah. So differences between LD score and Gremel. So Gremel builds off of a like mixed model type approach to thinking about the idea that the relationship between the phenotypes for a set of individuals and how similar a given pair of individuals is, if a phenotype is heritable, should be in some way proportional to how genetically related those individuals are, and so essentially uses an estimate from your individual level data of how genetically related they are and how that's tracking with their phenotypic relatedness via maximum likelihood approach. And so the kind of basic underlying model in terms of that there are SNPs with effects and there's some heritability term and essentially treating that as random is still there. Where the kind of the main thing that sneaks in terms of practical difference between the two is what this ends up meaning for rare variants and other structural variants, kind of things that aren't within the SNP data itself. And so that treatment of variants that is explained by things tagged outside, because LD score, we're reliant upon looking at what LD between SNPs are in either within our data or within some reference sample, versus in the Gremel approach, you can have some expectation that if people have a given degree of relatedness at their common variants, their rare variants probably have that same degree of genetic relatedness. And so the amount that you pick up that signal in GCTA is a bit clearer than how much you pick it up in an LD score approach. So there's not, kind of broadly speaking, on average, you will see somewhat higher SNP heritability estimates from GCTA than from LD score. And I don't think I have that slide in here, but there's a nice paper by Matt Stevens' lab, or I'm sorry, Matt Keller's lab, kind of doing direct comparisons in a number of simulation settings, including like whether or not the true causal variants are rare versus common and things like that, and different versions of fitting these models, and shows some of those average differences. Yeah, great question. All right. Right. A few other wrinkles to what actually happens in terms of fitting LD score regression. The first is that I've described this as a regression, but if you spend a little bit of time looking at what's in those, the equations that got us there, there's some definite heteroscedasticity in that expectation if we were to look at the expectation for the variance of those chi-square statistics. And if we run this on just all SNPs in a GWAS, we have correlated SNPs that are in our regression that would be equivalent to having correlated observations, and so we want to adjust for that. And so that's handled by at least a couple steps worth of iteratively weighting the regression. Similarly, because of that correlation between SNPs, analytic standard errors aren't going to be well behaved, also distributional assumptions. And so in practice, our standard errors for all the quantities out of LD score regression are reliant on block jackknife procedures. And then kind of the point we're just making is that the kind of universe of SNPs that have gone into this analysis, which really manifest in our choices about how to compute the LD scores, so both in terms of what set of SNPs are we going to include in our regression? So what set of chi-square statistics are we regressing on? And when we computed the LD scores for those SNPs, what set of SNPs did we sum over? Which doesn't necessarily need to be the same set of SNPs that we observed and used in our regression. And so given that slope that we get out of that is an estimate not directly of SNP heritability, but of a SNP heritability per SNP, so that A squared over M. To get to just a SNP heritability estimate, we do make an assumption about number of SNPs. And in practice, we make that assumption by kind of carving off the rare part of the variation so that we don't have to make as strong an assumption about what effect sizes look like as you start getting towards rare and rarer variants. There's still an assumption there. We'll talk about that in a second, but we at least make less strong assumptions by cutting that off. But because we've then trimmed that out of how we've gone about our modeling, it has essentially changed the universe of SNPs that we were talking about the SNP heritability explained by. And the last wrinkle here in terms of how this actually gets fit is that in practice, the default behavior in the software has been to exclude highly significant loci from this estimation. That does bias this, but does buy a lot lower variation in the estimates in a kind of classic bias variance trade-off sense. This probably isn't recommended behavior anymore, especially as sample sizes get larger and the number of highly significant loci continue to increase, but just something to be aware of in terms of default behavior of software implementations. Patrick, you have a look like you want to chime in on that.
No, I mean, it's more about it being an outlier than being high, right? But so you could write it so that if there's like a outlier, then maybe you find, but it's not very important.
Yeah, so the existing implementation actually has two different versions of this filter, and one is more outlier-y relative to sample size, and one is more just a flat cut. But yeah, indeed there's flexibility there. Yeah.
The comment about correlation between SNPs
Mm-hmm.
at the top. So this is a concern about two SNPs in the sample that are the same across people in terms of its genotype.
More or less, so the SNPs are correlated like in an LD sense within the GWAS sample. And so in talking about the expectation of their chi-square statistics, they're not independent observations. Like you would, if I told you that this SNP has this GWAS result and this is a neighboring SNP that's perfectly correlated with it, I haven't given you new information. And so in terms of constructing a regression where our observations are each a SNP out of the GWAS, we don't want to like double count that information from loading in SNPs that are perfectly correlated with each other.
Okay, but there'll be correlation, like by definition of LD, right? Sort of all the chi-square statistics within the graph will be non-independent.
Right.
Okay.
Yeah. So the treatment of that here is essentially SNPs get down-weighted proportional to their LD score, and so this is actually evident in the way these plots are constructed. So the regression weight here that is being referred to that shows higher weight for these low LD score SNPs as opposed to the higher LD score SNPs is that the SNPs with relatively lower LD are given proportionally higher weight in the regression process.
Got it. Okay. Thank you.
Too far. Here we go. We've got what, like 10 minutes to a break or something? I've got the time of presentation, but not the clock.
You have 20.
I've got 20, even better. Cool. Right. So yeah, so that bit about other implementation details was the last I was going to say about estimation, the estimation process, but I just wanted to reflag some important assumptions we've made and where they kind of interact with more practical realities. So the first is that tried to point out at the time that we make that strong assumption about those joint effect sizes of the best linear predictor betas being fully random in IID across SNPs. And that means that we're saying that there's not like local structure where all the SNPs in a given gene have higher effect sizes, or at least more likely to have higher effect sizes. We're saying we're going to ignore any correlation structure of that sort. where by calling them IID relative to having normalized our genotypes, we're making an assumption about how those effect sizes scale with allele frequency, so that essentially allele frequencies maintain constant per SNP variance explained as opposed to constant per allele variance explained. And because of all this discussion about kind of a universe of SNPs and only talking about heritability from common SNPs, all these assumptions are being made with respect to this kind of cryptically defined set of joint betas as opposed to necessarily true causal betas. So we've got that piece of assumption. We also have the assumption that our errors on our estimation of GWAS betas are behaving as we'd expect from OLS. And kind of the main wrinkle here increasingly is that more and more GWAS are at least considering a mixed model approach with the kind of use of a genetic relatedness matrix to help control for ancestry and not just BCs. And that constitutes a violation of this in that now kind of the explanatory power of that GRM affects the variance in our residuals, and so can lead to miscalibration of trying to apply LD score results to results from a GWAS that was done using a mixed model. We've already talked about the SNP selection issue in terms of kind of the weirdness of this estimand of SNP heritability. There's also our expected power or the efficiency of the estimates, both for the intercept and for the SNP heritability, but more so for the SNP heritability, are dependent upon the genetic architecture of the trait. So for the purposes of weighting the regression, we have to make that under a stronger assumption than getting to the regression model itself, in that we actually assume an infinitesimal model rather than simply just what those moments look like for mean and variance of the effect sizes. And because we're doing block jackknifes, which are blocks contiguously across the genome, if most of the genetic signal is concentrated in specific locations due to like just lesser polygenicity, then the distribution of that variation between blocks of a block jackknife is going to go up if there's more lumpiness to where that signal is. And so kind of less polygenic traits tend to have less stable estimation of all the quantities out of LD score regression. And then the last piece is for the separation between what's in our intercept as confounding and what's in our slope as signal. We have assumed that the amount of confounding in a given SNP has a constant variance that most importantly is unrelated to LD. It turns out that the unrelated LD is much more important than the constant variance piece, but that does leave some room for a signal that is actually confounding, for a signal to bleed back and forth essentially between our intercept estimate and our SNP heritability estimate under certain circumstances that we'll talk about more with the partitioning. Cool. So those caveats aside, what use do we get out of these things? So it's already come up that we've got the LD score intercept that can serve as an estimate of degree of confounding that's left in a GWAS, or if not confounding, then at least signal that we can't explain by our simple polygenic model. So kind of the simplest statement of this is that under a null hypothesis of no confounding and assuming all model assumptions met, then this intercept should be 1, so we can simply test that intercept coefficient against that null hypothesis. And under ideal circumstances for that for our scenario with the intercept, not only can we have that test for deviation from 1, that quantity overall is serving as an estimate of the variance of the chi-square statistic for null SNPs, essentially on the basis that for a SNP that is not tagging anything else, so has a LD score of 0, then our expectation for its chi-square is whatever this intercept is. And that kind of implies that if everything's behaving as expected, that this is the is proportional to how miscalibrated the standard errors in our GWAS were, and thus we could correct our standard errors for this residual unexplained inflation by multiplying standard errors by the square root of this intercept, which has been used to varying degrees in the literature. Certainly doesn't approach common practice everywhere, but is common practice in some places. But is at least a potentially useful way of thinking about something between just always taking the kind of raw GWAS result and going kind of the historical extreme of genomic control, where you kind of flatten for the average chi-square statistic overall, which will be very harsh if there is indeed true polygenicity. But that is relying heavily on believing that that intercept is actually correctly calibrated. The other way that we can look at this intercept is as a ratio. So in particular, because this intercept is estimating, if there is inflation, is estimating a inflation term that scales with sample size. Interpreting the intercept itself as a raw number is difficult because it is kind of related to this scaling variance term. And we may be interested not so much in kind of the absolute amount of variance from inflation, or like the absolute side of this A here, but more so how that relates to the overall amount of inflation we saw in our QQ plot, or in terms of how large our mean chi-square is. So the other commonly reported value is to take the intercept, subtract off its null expectation of 1 to leave just this quantity. And similarly, take the mean chi-square and subtract off that null piece to get essentially a ratio proportional statement of that total amount of inflation that's in the mean chi-square, how much of that inflation is not being is left over after we've regressed out the part that's proportional to LD. So this gets you a nice scaled 0 to 1 value that'll be more consistent across sample sizes and such in terms of having something interpretable. All right. So all those uses, let's look at at least a little bit of data on how these things behave. So I think the statement was already brought up earlier this week that Turkheimer's bold statement is that everything is heritable. And by and large, for LD score results across the ever-increasing number of GWASs that have been run, that looks largely true. So this is results from GWAS of thousands of traits out of the UK Biobank. This kind of collection of volunteer adults age 40 to 70, I believe, at the beginning of the study. 500,000 individuals in the UK who came in, gave biological samples, sat through a very extensive battery of behavioral measures, also linked data to their kind of hospital medical registries, and so has provided a wealth of phenotypes with a wealth of genetic data for 500,000 individuals, all genotyped, and so allows large empirical evaluation of across at least the set of things that somewhere like UK Biobank chose to measure, how methods behave and what distributions of results look like on average. And so I'm going to return to that set of results a few different times over the course of this talk and the other LD score talk. But so starting with kind of the SNP heritability piece, we see that kind of, at least for things that are well powered enough in terms of sample size and the biobank and case control numbers, the vast majority show at least some degree of SNP heritability, with the average kind of it being around 0.1, regardless of how you try and filter based on power availability. So the split here of the 3 parts of the QQ plot is relative to kind of confidence in given phenotypes based on their available sample size and some other measurement features. But so by and large, centered around 0.1, with the tail especially towards kind of easily measured anthropometric or biomedical measures, so things like height and weight, where measurement error is relatively limited. We might have kind of a prior expectations that the heritability is truly larger. That's at least a sense of when you get SNP heritability for a given trait at random, like what baseline expectation might look like. That's LD score SNP heritability. How does it compare to other heritabilities? So we've already talked about that it tends to be a bit lower than GCTA and why that is. It also tends to be more markedly lower than, say, existing twin or family study-based estimates for much the same reasons as GCTA in terms of what the actual estimand is for SNP heritability versus total heritability, and also how we're getting there. So the example I've got on the slide here is for a set of psychiatric phenotypes with the twin heritability estimated out of some large national registries on the orange bars, and the green bars here being LD score based estimates. And so you can see substantially lower, but still showing significant heritability for things we'd expect significant heritability for. And perhaps importantly, and not entirely kept track of here for some varying reasons about the phenotypes, but generally holding, and particularly in the simulation settings, considered, this is the paper I mentioned earlier from Matt Keller's group. Even if there are differences between what that LD score SNP heritability says and the values get from other methods, by and large, the trends in those estimates, if you look across the set of traits, will be consistent. So pretty much every method you can throw out SNP heritability will say height is highly heritable and is higher than, say, other more behavioral things. Maybe something like neuroticism's lower. So even if comparisons of those estimates between methods are difficult, it's at least still maintain some degree of meaning to make comparisons across different phenotypes from GWASs. Cool. All that was about just the vanilla piece of LD score that is just an intercept and a single statement about SNP heritability. And while those two parameters are super interesting and super useful, and I swear there's a reason I spend all day talking about them, we might be more interested in breaking up that heritability, and especially being able to model something more than just like, here's an average effect size across the genome. And so that was the thrust of getting into this second paper, where this is the original statement of that partitioning, but for purposes of kind of walking through how we get there, we're going to follow more closely with the generalization that came slightly later, led by Stephen Gazal here. And so I think, so you're looking at time here. We won't get fully through talking about the partitioning piece today. We'll pick up the rest of that tomorrow morning. But we'll at least try and get through kind of the initial idea of it, if not what the results tend to look like. So, as I said, we previously made the assumption that we have this single heritability per SNP that applies to all SNPs. But we can imagine, already kind of started to discuss, many scenarios in which that's not true and where we'd be interested in the ways in which it's not true. So perhaps wanting to make statements about whether there's more of that heritability per SNP that lives in coding regions versus the quote-unquote junk DNA that's elsewhere, or that are in kind of surrounding genes for genes that are expressed in certain tissues, or maybe we want to kind of play with the assumption that we made before about how effect size relates to allele frequency. And so all these things kind of give us a natural reason to want to extend the model to at least do some degree of modeling variability across SNPs and what this heritability per SNP looks like. And so to return to our cartoon example, how might we accomplish this? Well, so we can imagine that in our toy genome here, that where we previously just had a set of SNPs and their LD blocks, and defined this LD score that essentially amounts to the count or the sum over how much LD each of these SNPs have to their surrounding SNPs. In this toy genome, there are known differences between these SNPs. So we have perhaps a chunk of SNPs that are in blue here now that are coding. So maybe this is an exon for a gene that happens to live over here and spans a little bit across the LD blocks. Maybe over here we have a DHS site, so DNase hypersensitivity site, which is a particular measure of like accessibility of the DNA to some of the proteins, getting in and interacting with the DNA versus being tightly packed in chromatin and not really doing anything maybe. So we might be interested in kind of treating those SNPs differently in our model. So what we can do is we can take our LD score and simply say, well, what if we only count, in looking at LD to other SNPs, we only count LD to SNPs in one of these categories. So only count LD to the DHS SNPs. So all the SNPs in these LD block are related to this subset of SNPs that are actually annotated as being part of the DHS site. Similarly for coding, we've got some SNPs over here where, like, SNP here is at least in LD with this one, so maybe we'll expect to see any causal coding effect that's happening here still show up in the GWAS for the SNP as well via the LD. And so we can kind of split up our idea of LD into LD to different categories. And so the generalization of this, in terms of trying to think of this in terms of the math, is to say that the variance of those joint not quite causal effect sizes, instead of being flat across the genome, is now summed across some set of annotations related to the annotation value for that SNP. So we've got A is an annotation of the DEEF annotation out of a set of annotations for SNP-J, is this term here. And then that there is some variance parameter affecting kind of the total quantity of variance in those effect sizes that is the variance related to this particular dimension of the annotation. So if we have just a kind of disjoint set of binary annotations, then this can turn into a statement of that these taus are essentially the heritability of a per SNP for a particular class of SNPs. But in this general sense, where both this annotation can be continuous and a given SNP can be in multiple overlapping categories, multiple values, you get a generalized statement here that you have some contribution per unit of this annotation of those taus. But those are still compatible with saying that there's the total relationship once you sum across SNPs and across those annotations that gets you back to the total SNP heritability of our overall model.
So annotations, is that like the coding score? Or what is it?
Yeah, so if we take that toy example where we just had, like, there was some set of SNPs that we called coding, then this annotation would be a 0-1 indicator for whether SNP-J is one of those coding SNPs, and then what we're moving towards is being able to estimate this tau that is the kind of contribution to per SNP heritability that is different between essentially that variance in per SNP heritability for SNPs where this annotation is 1, so where they're coding will include this tau, versus if this is 0, then it will not include this tau. And the statement will pretty much always include that there, amongst this set of annotations, is one baseline annotation that is simply an annotation that contains all SNPs that are part of the model. So there's always some variance present in the model for across the set of annotations. Cool. So if we take that new statement about what our effect sizes look like across SNPs and plug it back into where we were before about effect size per SNP and how that relates to what we see in the GWAS betas, and walk through expectations again, we get essentially the same kind of pieces around stratification and estimation error, but now have that this expectation about the variance of these betas is being taken over by this expression in terms of the annotations. And we're going to, because we now have this additional summation term where we're having to deal with summation across the different annotations that are in our model, what that leaves us with is, so we've got these taus, and we can now break up that LD score to surrounding SNPs to define an LD score of SNP-J to all its neighbors K, that's part of this, for a given dimension. So it's based on where that LD is being affected by the value of the annotation for SNP-K. So we only count LD to SNP-K if it's a coding SNP, for example. So that's the kind of expression of that here, that the SNP-K and the LD that we sum across. And that leaves us with another regression-y looking thing where we just have to deal with multiple regression now for our multiple LD scores that correspond to these annotations. And I think we're at time for the break by like 2 minutes. So I think you're going to break there. We'll talk next time about kind of what actual results from partitioning have looked like and examples of use of that, and then get into genetic correlation. Any questions before we cut off there for now? Yeah.
I just want to confirm in the partition case, we assume that stratification is constant across annotations, if that makes sense.
Yes. So we're assuming that previously the statement was that the stratification is not related to the LD score, and now that's the stratification is not related to the LD score for any of our annotations.
Okay.
Though, kind of to preview where this is going, we may find that there are annotations that essentially are helping account for places where we might expect that to break. So in particular, annotations that start getting increasingly close to annotations of like degree of variation there is between populations at given sites. Yeah.
Okay.
Anything else? Great. Thanks.